Today, we'll be discussing management strategies for plant viruses. Management strategies for plant viruses focus on prevention and suppression, as there's no chemical cures for an infected plant. A successful management plan integrates multiple conventional and cutting-edge biotechnological approaches. So, management strategies include cultural and sanitary practices, modern biotechnological strategies, implementing an integrated approach. So, first we'll start with the cultural practices. Cultural practices are low tech methods, create an unfavorable environment for viruses and vectors. Cultural practices include sanitation, crop rotation, vector control, then adjusting planting and spacing. Viruses can persist on tools, debris, and weeds. So disinfect equipments with 10% bleach solution and remove infected plants and crop debris to eliminate sources of the virus. Then alternating host crops with non-host crops disrupt the disease cycle and prevents the buildup of viral infections in the soil. Then vector control, since many viruses are spread by insects like aphids and whiteflies, controlling these vectors is critical. So this can be done with insecticides, reflective mulches, or by planting non-host barriers crops around the field. Then manipulating planting dates can help plant avoid peak vector population. So proper plant spacing increases air circulation, then reduces humidity and slows the spread of the foliar viruses. Next, use of virus-free planting material. So using clean certified stock from the start is one of the most effective preventive measures for controlling the viral diseases. So national and local program test and certify seeds, tubers, and other propagative material to ensure they are free of known viruses. Then next are modern biotechnological strategies. So in uh, breeding for resistance is an effective and economical long-term strategies. So we will discuss two methods under modern biotechnological strategies: host plant resistance and biological control. Host plant resistance include conventional breeding, RNA interference, then CRISPR or Cas9 genome editing. Plant breeders use traditional methods to develop new cultivars with natural resistance genes to viruses. Then RNA interference. This is a natural plant defense system and can be leveraged to create genetically modified crops resistant to specific viruses. So the plant is engineered to produce small RNA molecules that interfere with the virus's ability to replicate. And in CRISPR or Cas9 genome editing, this is advanced gene editing technology and allows for precise modification of plants' genome. It can be used to target and inactivate viral genomes or to mutate host genes that viruses rely on, thereby conferring resistance. Next, biological controls. This is eco-friendly approach and it uses other organisms to manage viral diseases, often by controlling their vectors. So, certain strains of bacteria and fungi act as antagonists to pathogens or induce systemic resistance in plants, strengthening their natural defenses. In some cases, viruses that infect insect vector, such as baculoviruses for caterpillars, are used to control the spread of plant viruses. Another method is crop protection. Here, inoculating a plant with mild strain of virus can sometimes protect it from the more severe strains. Through this method, it carries risk. And last approach is integrated approach. For sustainable and long-term control, the most effective approach is an integrated disease management program, IDM, that combines multiple strategies like Start clean, use resistant cultivars, monitor regularly, practice good sanitation, control vectors, utilize advanced diagnostics, plant certified virus free seeds and propagative material uh, is to start clean. Then select varieties known to possess genetic resistance or tolerance to local viral threats. Then inspect fields frequently to identify and rogue infected plants and detect vector outbreaks early. Remove and destroy infected plant material and disinfect tools to prevent mechanical transmission. And deploy barrier crops or use targeted insecticides when vector population pose a threat. Thank you.